So I'm speaking on behalf of Zumedix, and we are a Seattle-based company that's formed around a novel technology for topical antibiotics. And this was developed by my co-founder, Dr. Eric Brown. He's a world-renowned microbiologist at McMaster University outside of Toronto, and he found while researching a new antibiotic that the drug worked much better in an animal than it did in cell culture. And then he, puzzled by this, he tried to figure out why that was, and after a lot of research, he figured out that the mediating factor was physiologic bicarbonate. And then he underwent an extensive testing looking at several antibiotics and bacteria, and to try and see which that it may be that existing antibiotics would be enhanced. And actually they were. It turns out that there's many, not all, but many families of antibiotics that are enhanced, and they kill many types of bacteria. So we believe this is a transformative technology where bicarbonate can both enhance the effectiveness of existing antibiotics as well as increase their spectrum. It addresses several medical unmet needs in many, because this can be used in many indications, not just the eye, but any place you can use a topical antibiotic. Our target market, as I mentioned, is topical, and the technology is suited for the 505(b)(2) pathway because it's all approved compounds that we're using. And our first product, our lead product, is Zumedix 001, which is an azithromycin bicarbonate formulation. As you all know, antibiotic resistance is on the rise. Increasingly, keratitis isolates as well as isolates from otitis contain more and more virulent hospital-borne resistant species. And additionally, existing treatments are really ill-suited for polymicrobial infections of more than one kind of bacteria. There's also a real lack of innovation in this field. Most of the antibiotics we use are based on technology that was developed about 50 years ago. So that's why there's so many generics out there and not so many new compounds. So we feel that the topical antibiotic market is ready for a new leader. And so Zumedix is going to be that leader, and there's many indications that we can use this drug: the eye, the ear, the skin, and really any mucosal place where there's an infection. And there's many antibiotics that we can enhance. So we can enhance the aminoglycosides, fluoroquinolones, macrolides, polymyxins. We can also enhance antiseptics. So if you think about how many antiseptics are used in the hospitals and things like that. We've shown that we can convert FDA-approved non-antibiotic drugs into antibiotics, and we think that we can also develop new antibiotic classes with this technology. This is a little cartoon that shows you the let's see, that's okay. That shows you the mechanism of action. It turns out that that BICARB increases the membrane potential across the bacterial cell wall. And this increases the negative charge within the bacteria. And it allows antibiotics like here, this is azithromycin, which is positively charged. It draws those antibiotics into the cell. So we get higher efficacy because of that. And we've shown that many of these, we can see more antibiotic in the cells when we look. So the next few slides are going to show this technology in action. So methicillin-resistant staph, or I should say Staph aureus, about a third of it is MRSA. And 80% of our MRSA is resistant to multiple, at least three antibiotic classes. 85% of MRSA is resistant to azithromycin, for example. And just for reference, azithromycin susceptibility is considered anything at 4 micrograms or lower. So if it's above 4, it's not susceptible. And so on this graph here on the right, you'll see that we've looked at over 100 MRSA species that were clinically derived, many from the eye. And we found that when you use azithromycin alone, so that's the column on the left, it's virtually, it's resistant. You have to use, it's just not an effective drug for MRSA. But when we add, when you add bicarbonate, you can see that we dramatically lowered the MICs and all within the susceptible range. So we've increased the efficacy of azithromycin in MRSA. Similarly, but surprisingly, we found the same results with Pseudomonas. So you don't expect azithromycin to treat Pseudomonas, but when we add bicarb, it does. So now we've changed azithromycin into a more effective drug and a broad spectrum drug. And then to kind of give this some grounding, we look at all the drugs we use topically, or most of them here, and you can see with the same isolates, we did the same testing. And you can see our compound on the far right. And here, that we have basically leapfrogged the entire market on topical antibiotics. So we have a much more effective drug than all of these drugs. And then we did this an in vivo model in skin, and this is a standard skin infection model using Pseudomonas. And what we found is that we can lower we count the number of bacteria, and we can lower the bacteria rate by over 3 log units with our combination. And 3 log units is really considered a treatment. So anything less than 3 log units, it's considered not fully treated. So we've been able to show this in vivo as well. Our development path is relatively simple. These are well-beaten paths for developing this for skin, for eye, for ear. With the 505(b)(2) pathway, we think we can get to phase 2, completion of phase 2 for about $3 million. Basically, we feel that Zumedix is well positioned for success with this groundbreaking technology that improves the efficacy and spectrum of existing and potentially new antibiotics. It's been proven in vitro and in vivo now. We have multiple formulations and potential indications. It's a rapid, inexpensive, and low-risk development pathway, and we are seeking strategic partners to help advance this technology to the clinic. Thank you.